Namaste students, I am Jayoti Dr. Achis Mohammed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapeeth, Women's University, Jaipur. Today I am going to discuss about the topic organization of prokaryotic cell. This topic belongs to the fundamental of microbiology. This topic is in Vidyapeeth, uh, second semester. So uh, there are two types of cell. Uh, the first one is prokaryotic and the second one is eukaryotic cell. Cell is the basic unit of life, and on the basis of the presence of the nucleus and nuclear membrane, there are two types of cells. First, prokaryotic means they have primitive nucleus, means the nucleus is not present and it is not observed in the cell. And eukaryotic means the nucleus is very well developed and it is easily observed. So if we talk about the different cells, different microorganisms which are present on the different smallest structures, then you can say that the size of the living system, the atom, first thing, the atom, the smallest thing, is 0.1 nanometer, and the amino acids are of 1 nanometer. They have a size of 1 nanometer. From that amino acid, proteins are larger, which have a size of the 10 nanometer, and viruses are larger than the proteins, which have a size of 100 nanometers, approximately 10 to 100 nanometer. And from these viruses, the chloroplast is larger, which is 1 micrometer in size. And from this chloroplast, some bacteria are larger, most of the bacteria are larger, which have the size of the 1 1 to 10 micrometer, and some of them may also have some larger size. And from that bacterial cell and some microorganism like fungi have a size of larger 20, and the plant and animal cell have a diameter of 100 micrometer. The human head is expected to have a diameter of more than 100 micrometer, and the frog head is also larger, which is the 1 millimeter, and ant is approximately 1 centimeter, and mouse is approximately 0.5, larger than that, maybe approximately 15 centimeter, and from that the human will range from a 1 meter to several meters, for 1 meter, 2 meter, maybe any foot, 5 foot, 6 foot. And the blue whale is the largest, which may have a 10 meter. So these are the different sizes, and the different scales on which you can say that the eukaryotes have size of 10 is for 3 millimeter, and prokaryotes are larger than them, viruses are larger than eukaryotes, and proteins are larger than the viruses, and atoms are smallest structure in the living system. So the cellular level of the organism, the living things are constructed of the cells, and all the living cells may be unicellular and multicellular. Cell structure is have some characteristics, and they are very small and exchange the materials from the surroundings. So there was a theory which is known as the cell theory. What does the cell theory says? Cell theory says that all the organisms are composed of the one or more cells. Cells are the basic unit of structure and function of the organism. Basic unit. And the third thing is all cells will arise from the pre-existing cells. They will not arise by themselves, but they will arise by the pre-existing cells. So this is the cell theory. Uh, there are two basic types of cell: prokaryotes, which do not have a membrane-bound nucleus. And the example include the bacteria and the archaebacteria. The second type of cell is the eukaryotes, which have a membrane-bound nucleus, uh, which include the algae, fungi, protozoa, plants, and animals. If we talk about the prokaryotes have two organisms, bacteria and the archaebacteria. So we will discuss about the structure, organization of this bacterial cell. And the bacteria, bacteria have some chemical composition. Bacterial cell have some chemical composition which include water, which is approximately 70%. DNA is approximately 30%, and RNA derived is composed of 30%, and DNA from this derived 5% is there. 5% molecule is composed of the DNA, and RNA is approximately 12%. Proteins are approximately 70% of the derived. From this 30% derived, DNA has 5%, RNA has 12%, proteins have 70%. And similarly, ribosomes, protein particles, enzymes, some surface structures are also there. And polysaccharides have approximately 5% contribution in the drive, and lipids have 6% contribution in the drive of the phospholipids have 4% contribution in the drive of the uh, side. So how bacteria will reproduce, and how they can reproduce. They have a reproduction system by binary fission. The cell will divide into two equal parts by the process of the binary fission. We talk about the structure of the prokaryotic cells. The prokaryotes have some important cell structure like appendages. The prokaryotic structure is divided into some parts. The first one is appendages, means the part of the cell which exists outside the cell and which provide support to the cell, include flagella, villi, fimri. So these are the cell appendages. Next, the cell envelope, which is composed of the glycocalyx and cell wall. So second part, the third part is cytoplasm. Cytoplasm means the part which exists inside the cell membrane, or the part which exists inside the cell wall, which is known as the cytoplasm. So cytoplasm has a various number of cell organelles and has important, most important part. Has ribosomes, granules, nucleoles. So all the organelles are present in the cytoplasm. But in the cytoplasm, there are some prokaryotic genetics. What is the genetics? Genetical organelles in the prokaryotes. The first one is nucleus. Nucleus is the region of the cytoplasm where chromosomal DNA is located, and the DNA is singular and circular chromosome, and few or no proteins united are present there. The plasmid is also there in the cytoplasm. Plasmid is also there, and the small structure piece of the chromosome genetic material is known as the plasmid. Because the prokaryotes and some bacterial cells have a DNA molecule which is present. A chromosome is there, but beside this chromosome, an extra chromosomal DNA is also found, which help them help the bacteria in not only protection but also it help in the reproduction. It can provide genetic information and antibiotic resistance and resilience factor. So these factors are related to the plasmids. If we talk about the important part which differentiate between the different type of bacteria, then it is cell wall. From the peptidoglycan, there is a cell wall which differentiate type of bacteria, which is the gram positive and the gram negative. The peptidoglycan inside is very similar. If we go further, the bacterial cells are divided into two major classes. One which gram positive and the second one is gram negative. So gram negative and gram positive, what is the difference between the gram negative and gram positive? The peptidoglycan layer is thicker in the gram positive bacteria and it is thinner in the bacteria. That is the major difference. If we talk about the cellular shapes of the bacterial cells, the different shapes can be the cellular structure is round shape of then the coccus. If one coccus is there, it is known as the coccus, monococcus. And if two coccus mix together, then it is known as the diplococcus, diplococci. And three or four coccus cell bound together, and they are known as the tetrads. Tetrads. And if two tetrads mix together, then they are known as the sarcina. Okay, sarcina. And if the focus bound together like a grapes, the focus structure bound together like a grapes, then they are known as staphylococcus, one single branch, known as the staphylococcus, like a branch of grapes. So they are known as the staphylococcus. But if they combine, they form a colony in chain, then they are known as the streptococcus. So from this we can say that the bacteria cell exist in the colonies. They exist in colonies. They can be said that they are Sarcina, Staphylococcus, and Streptococcus. The second stage which can arise in the bacteria is root cell bacillus. If they are the cell in the root cell, then they are known as the bacillus. If one bacillus, one cell is there, it is known as the monobacillus. If two cells are there, they are known as the diplobacillus. And similar thing are which are we were discussing about the root cell. If they are found in the chain, they are known as the streptobacillus, and if they are in the bunch like the grapes, and they are known as the Staphylococcus. Some more structures can also be found, such as the spiral type. The cell may have some spiral, which are known as the spiral type cell. Spiral, spirulina, and some of them can also found spiral, spiral way also, which are known as spirochetes. Okay. Some coma shape can also be found, which are the examples of the vibrio coma shape, which are the example of the vibrio. So uh, these can be found. These structures can be found in all the bacterial organisms. So I hope the students have better understand all these things. Uh, so here I must conclude here that the bacteria have a cell which is composed of some three parts. The outer part have some cell appendages which include the cell uh, appendages like cell, flagella, villi, and familia. So cell cellular parts. Second compartment is cell envelope. So in the bacteria, the outermost layer with glycocalyx and cell wall. So this is the part which protect bacteria from the separate bacteria from the outer environment. Inside this cell wall, there are some another covering which is known as the cell membrane, and inside the cell membrane, the third part exists which is known as the cell cytoplasm. All the organelles of bacteria and the genetic organization is contained inside the cytoplasm. The DNA, RNA, proteins are present inside the cytoplasm in bacteria, and all the organelles present inside the bacteria, whatever like ribosomes, so all type of bacteria. In the next video, we will also discuss about the cellular compartments of the bacteria separately in different lectures. So my lecture is over here. If you have any query, you can ask me. Uh, Thank you very much.